Okay, I think this will be the last section of this. So organic molecules again, last bit. What we're looking at here is focusing on the components of cells, of things you find in cells, either structural units or things that, molecules that we use to make things happen in the cell. Okay, so I'm outside again, storms have passed for today, and I'm enjoying the outside. So you may hear a little breeze on the mic there. I apologize for that if that's disturbing. Sorry. Hopefully it's not too bad. Kind of have my back somewhat to the breeze, so hopefully it'll protect it. Carbohydrates. These are organic compounds that contain carbon, hydrogen, and oxygen. And so we have, do I have pictures of those? I was lucky earlier and had them. No, that's the fatty acids. Okay. So we've talked about this back, I think, second chapter of the book. We took a little detour and I gave you a little teaser of this. We talked about glucose, 6-carbon structure. We talked about fructose being a 6-carbon structure, having the formula, both of them having the formula C6H12. That's a really sloppy C. And with this background, I'm not sure the red is very good. Okay, so that's a monosaccharide. Okay, glucose, fructose, same structure. They have the carbons are arranged differently. There are diagrams in your book. You can look those up. I pointed them out to you before. Let me find something else. Okay, a disaccharide, if you take these and hook them together, take a fructose and a glucose and hook that together, you're going to get a C12 H22 O11. Okay, and that is sucrose. So if you look at that really close, look at the difference between two of these structures, or well, two of these formulae, and this formula. What are you missing? If you were to double this, you would have C12H24O12. So what are we missing? Two hydrogens and an oxygen, right? What does that make? So putting two glucose together, just like our proteins, okay, gets us a water. Condensation synthesis was a term used last time. Another term that I was taught, actually, I was thinking last time when I read that off the slide, didn't sound familiar. Dehydration synthesis. Okay, condensation, dehydration. Anyway, you pull the water out of it, condense the water out of it. We get 3 or more sugars hooked together, glucose, fructose, some monosaccharides, 3 or more monosaccharides hooked together. We get a poly. Poly means many again, more than 2. And then we get the potential for these really large molecules down here, like starch and glycogen. And again, you can see these in your book. They are made up of glucose. Okay, both of them. Both of them made up of glucose. They're just, the glucose molecules are bonded to each other in a different pattern, and that makes the larger macromolecule, the starch and the glycogen, have different properties. So your starch is the storage molecule that plants use to store their excess glucose. So they photosynthesize, they get this extra glucose, and they have to burn some of it in cellular respiration just like we do. They've got mitochondria, they're going to oxidize that, burn it, okay, and they're going to get energy out of it, but they make more than they need. which is great for us because we can eat them and get their extra energy because they will store it in places like their root or something like carrots, turnips, radishes, the root crops. Some will store it in a modified stem, like a potato is a modified stem. Then you have storage in fruit. Okay. A lot of things that you think of as vegetables, like tomatoes, peppers, um, those are actually fruits, botanically speaking. Okay. Because anytime you have the seed containing structure of the plant, that's actually the fruit, the fruiting body. So you have, cantaloupe and watermelon and green beans. You got the seed inside those green beans and peas and corn, even corn's a grain. That's a seed. Okay. So you're eating technically fruit. Most of the stuff you grow in a garden is technically fruit. If you're eating the actual plant structure, like I'd mentioned, where you're eating the root, the stem, the leaf, okay, then you're eating a vegetable. Botanically speaking, now, anybody who's taken culinary arts and a chef's like, yeah, well, it's all about the use. And I'm like, well, yeah, okay, but I'm gonna speak from the scientist, but botanist type perspective here, uh, and I always do. So almost always. So fruit, okay, a lot of starch stored there, and a little fructose too. The sweeter the fruits, the vegetables are often starchier. That's what we think of the fruit that we use culinarily as a vegetable are usually starchier, and the ones that have more fructose as opposed to starch are what we refer to as the fruits. So yeah, okay. But anyway, we benefit from the plant's ability to do that. Animals, we also have, we ingest more than we need and we store it. Say, well, that's fat. Yeah, well, it's glycogen first. Okay. And then we get a manufacture of adipose tissue and stuff like that. But it's the glycogen is the glucose molecules that are uh, readily accessed for energy when we are between meals. We need a little extra uh, access to some glucose because yeah, we either didn't eat enough or we're not to the next meal yet, or we've gone and worked hard and exercised and burned off stuff faster than we would have normally, that type of thing. So these are the two storage molecules um, for plants and animals. And then my biology students are learning have learned that like fungi store their extra glucose as glycogen as well. Fungal cells are more like animal cells than they are plant cells. Really, the only thing that kind of makes them plant cell like is that they've got a cell wall. Uh, Otherwise, they're many ways they're much more like an animal cell. So let me erase so I can move on to the next slide. Okay. Now, the third large molecule, macromolecule that is made up of glucose is cellulose. It's a polysaccharide, lots and lots of glucose molecules. I don't know that your book has a diagram showing how these molecules, these glucose molecules, are bonded together in yet a different pattern from the other two to make this particular macromolecule. It might. In past semesters, I have never, and I moved outside. I didn't bring my book out with me because it's a little damp out here, but I didn't think I needed it at this point because I'm not referring to the structures on the back of the book and that sort of thing. But I don't believe that I have ever really stressed showing students in previous semesters the differences in cellulose. I did want people to see the difference in structure between glucose used bonded together for starch and that bonded together for glycogen. I don't quiz you over that. You don't have to identify those structures on the test, so don't worry about that. I just wanted you to understand that yes, we can bond the same things together in different ways and get different stuff. Uh, and I think I figured that if you can see that with starch and glucose, then you understand there's another way for the cellulose. So kind of didn't worry about that. What cellulose does is it's a chief constituent component of the plant's cell wall. Uh, that's not necessarily anything that you need to know going into human anatomy and physiology, um, even micro, going into health sciences for humans. But uh, where cellulose is important in from the human standpoint is that is our fiber. So when we eat plants high in cellulose, or parts of the plants that are high in cellulose, which we often don't. I mean, yeah, fruit has some fiber, things have fiber in it, but the really high ones are like celery stalks, okay, and things like that. Really high cellulose content cells, we have trouble digesting, and they can easily give us indigestion. They can easily cause us to have a lot of gas, because we don't have the enzymes to break them down. And so uh, we don't really have the enzymes to break cellulose down. That's why it's fiber, stays as fiber in our intestines and kind of cleans us out, scrubs us out, collects particles of things that have collected on the sides of the intestinal walls and or walls, membrane. It's not really a wall, but it's what we refer to it as anyway. And pulls and cleans that stuff out. So it's why we do need fiber. It provides a base for the bowel to build on, uh, so that you have a solid bowel movement. It, uh, however, does break down if it travels, especially if it travels slowly through the system. It breaks down from bacteria called methanogens. Uh, at least that's one of the major groups of our normal flora that act on it. And they try to help us out kind of by breaking it down a little bit. They get some energy out of it for themselves because they can dissolve it. But their byproduct is methane gas. So that's why we get gas from having a lot of cellulose in the diet. Okay. So, uh, dietary aspect is where you're going to, run into cellulose and kind of, I guess, need to know its basic structure to some extent. But the other molecules we're talking about here are going to come more into play as you have A and P. Okay. Lipids, I already talked about them a little bit previously in this section in another presentation. Um, Lipids are fats and oils. Those were the two I talked about. Also steroids. Okay. Um, this is an organic compound that's insoluble in water. Okay. Hydrophobic. Okay. They don't like water, won't dissociate in water, will bond to themselves or cling to themselves rather than dissociating in water. Uh, so steroids, they were talking about cholesterol, actually. Steroid. Cholesterol gets a bad rap, but I think I've got a little blurb on it here, another slide. So fat is an organic molecule that contains glycerol and fatty acids, and is found on our adipose tissue. And oils, and that fat is animals, okay? I say us, and it's, well, humans, because this is kind of focused on humans, but all animals. Okay, so we eat the fat of other animals or use the fat of other animals like butter and lard and tallow. Okay, that's what uh, we're eating or we're using to cook. Is that the oils are plant origin and they're usually liquid in consistency at room temperature because they're unsaturated, as I explained previously. And here we have a glycerol and 3 fatty acids in a chain. Okay, so picture of that. Okay, this is your triglyceride. All right, and the tri comes from that term. Okay, so you have a glycerol, 3 fatty acids, a condensation synthesis puts that all together to form the triglyceride, and you get 3 water molecules off of that because it's 3 different condensation, uh, 3 different condensation reactions to hook those together right there and there and there. And you see the blue highlighted atoms are what are going to be released so that the bond can form there and there and there. And that's going to leave us with 6 hydrogens and 3 oxygens to form up some molecules of water. As said previously, saturated fatty acids have no double bonds, and unsaturated fatty acids have at least one double bond. Let's go explore that. So these don't look like we've got any double bonds between the carbons. So, well, here's what, yeah, but that's that carboxyl group, and that doesn't count. We're talking about between carbons, double bonds between carbons. These are all saturated. Oh, look right there. There's an unsaturated. So this fatty acid chain is a different structure, okay, than this one. Different number. That one, same number, I think, is that one. Nope, they've all got different numbers. Okay, so but also double bond right there, and that makes it unsaturated. So phospholipids. Okay, so this is a lipid that has a phosphorus attached to it, phosphorus group. It's actually two lipid chains. And this is an important molecule because it is what forms our cell membrane, or sometimes referred to as plasma membrane. And of course, hopefully you've learned and remember from biology class that that is a double layer, okay, enclosing the cell, contains a phosphate group, has a hydrophilic head that likes water, and two hydrophobic tails that don't like water. Okay, so we draw this compound like this all the time. Very stick figure version, excuse me, of that is going to be, oh, I've got yellow still, show up against the black, maybe not so good against the white. Okay. All right. Here, over here is what it actually looks like. Molecular structure. Okay. The chains, these are the fatty acid chains, and then this is your phosphorus group here, okay, with stuff attached to it. For the head, this is called the head right here, head, and the tails. Okay. I think my next picture, I think I have a picture on the next slide that is, yeah, this is what I want. So I don't have to draw it out. And I won't make you draw it on the test because this next test is all multiple choice. So what you have is, here's the heads and the tails. And notice how they flip in toward each other. So water, we've got water in the blood. Blood's mostly water. Outside of the cell, the interstitial fluid, the fluid in between the cells, is mostly composed of water. In the interior of the cell, cytoplasm is still largely water. Okay, water solutions. These are solutions of things dissolved in mostly water. So the tails don't want to have anything to do with that water because they are afraid of water. They're hydrophobic, water fearing. And so getting these tails to flip inwards like that makes them happier, and these heads are the things coming into contact with the water. So this is your basic phospholipid bilayer in the plasma membrane. It's not this simple. Obviously, we've got other things embedded in the membrane. Okay. And I don't think I go into that, do I? No. Some semesters I do, but we won't this semester. Okay, let me get back to, wow. Okay, I kind of reset. Okay, so what I am going to say about this structure here then is that you have embedded in between these phospholipids, and some of them will bulge out, but beyond, they've got carbon chains, some of them attached to them, some of them don't. It's carbon chain. I could just draw an R there, right? Now that you know that, can't do that for my biology students. We're going, what? What's that R? Okay. If it's got a carbon chain attached to it, it's called the glycoprotein. And glycoproteins, and you can also have some of these phospholipids with that type of structure too, called glycolipids. And those are not terms you need to know right now for this class. You probably will talk about them in A&P. What these do, though, is they give the cell surface its identity, so that and that identity is unique to you. And that way, it's part of your body's immune system. It's part of your body being able to recognize a foreign substance. Your immune system sees this on the outside. Your immune cells recognize these glycoprotein and glycolipid patterns on the outside of your cell and say, ah, that belongs here. I shouldn't attack it. Unless you have an autoimmune disease, and then for some reason that doesn't work. Okay. Um, Other proteins that don't have those uh, are proteins that allow large particles to pass through the cell membrane here. Um, so water soluble substances can go, diffuse osmose, okay, through into the cell and also can go out of the cell. But some things that are charged particles and things can get tangled up here in these tails, okay? The fatty acid chains will grab onto them, hold onto them, you know, chemically. And so those things have to be transported through some type of protein, either a carrier protein or a channel protein, some type of protein. And then also things that are too large to osmose have to be transported in through and they can sometimes go through a channel or carrier protein. Things that are really, really large, however, can't pass through the membrane. They have to be taken in through endocytosis, where the membrane actually forms a vesicle around or vacuole around the substance itself and pulls it in. And that's how virus particles get in. Okay. They do bind to a site, um, and usually it's maybe probably a protein binding site, but instead of using that protein to travel through, because the protein changes shape and like carries it um, across, what need to stop? Okay, um, we get this folding of the membrane around. Okay, so that that aspect of the cell membrane is not something I'm going to quiz you over. I kind of want you to have heard it before you get to A and P. Um, and again, it shows you how some of these organic molecules, so organic particles, okay, molecules that we've been talking about, uh, are important in functioning in our system. Okay, so that's uh, the whole point of this end here is to show you how all this chemistry we've been in all semester, all year, well, semester, yeah, all year so far, all semester adds up to apply to a living being and a human in particular, and will help you hopefully understand structures and functions of physiology, because your A&P class is not going to be just about memorizing bones and muscle tissues, though it's largely that. You're going to have to learn some physiology. You're going to have to learn how things travel through the system and how mechanisms are turned on and off. And that's a lot of these proteins and things here. So understanding somewhat of their structure and basic uh, qualities hopefully will make more sense to you. And if anything, as you get into these classes where it's lots and lots of memorization like A&P and micro, anything that helps you feel less lost and or have a familiarity with something you heard about. It's like, oh, I've heard about that before. I know what that does, or I know how that's made up of it. It, for me at least, it makes it less anxious. I have less anxiety about learning about it because I kind of feel like it's an old friend. I already know it a little bit. Okay, so that's really pretty much uh, the purpose of this last bit, or this whole class, is to get to this last bit here and show you how it kind of ties together into an organism. So the steroids, yes, cholesterol gets a bad rap because of what it can do to us as far as clogging up our circulatory system, but we need it. Back to this picture here. We have something called a blood-brain barrier, and what cholesterol does is, along with those proteins I was talking about being in here, we can also get cholesterol. Cholesterol is embedded in the cell membrane. So some cell membranes, we don't want to have a lot of cholesterol because we want them to be a little bit more fluid, but we want them to have some. Every cell membrane needs to have a certain amount of cholesterol because it's one of the things that helps the cell control, reduces the permeability of the membrane. It helps the cell be better able to control how much is going in and out. Uh, the blood-brain barrier has a lot of cholesterol. That particular membrane has a lot of cholesterol in it, and it needs to because you don't want things crossing over that just by random diffusion. Okay, osmosis. Uh, there are a lot of things that we ingest that go very quickly. Uh, uh, will go very quickly from the mouth to the brain. They digest. The digestion starts in the mouth. Breakdown starts in the mouth, and they get absorbed through skin or through the linings. Uh, and so there are foods, chemicals in our foods, and things that we eat that until they're broken down by our digestive system, they're toxic to our brain. They're deadly even. Uh, they have to be metabolized, and after they're metabolized, they're great. They're fine. Have components we need. Um, but we gotta that blood-brain barrier there is to make sure that those components don't get to the brain before they've been metabolized properly. Um, the one thing that the blood-brain barrier and its amount of cholesterol doesn't seem to prevent from getting there very quickly is alcohol. Okay, uh, and this is why. If you've ever wondered why certain drugs are not supposed to be taken with alcohol, this is why. Because prescription drugs are definitely in the family of chemicals that need to be metabolized before they travel through the body in many cases. The metabolizing of them breaks them down, unless, of course, you're injecting something directly into the bloodstream, and then that's a different category altogether. But a lot of what we take by mouth needs to be metabolized. And alcohol is a solvent. Okay, it's an organic solvent. Water is a universal solvent, but things that don't dissolve in water will dissolve in alcohol. And it can carry substances across membranes very quickly. Uh, and so that's the danger. If you had never understood, okay, the danger of why alcohol mixed with certain chemicals is a really, really bad thing, is alcohol in itself, getting across the blood-brain barrier in large quantities uh, is deadly, and the wrong types of alcohol. If you straight ethyl alcohol, pure ethyl alcohol, is a very, very dangerous thing. We water it down. What we typically ingest is not 100%, and even some of the higher percentages are probably questionable about whether we should be ingesting them or not. But um, beers are really pretty actually low percentage. Wines, what, 14%, something like that. So uh, your percentages are mostly water. Okay. But then you have alcohols other than ethanol, and they're toxic in smaller quantities. Okay. So uh, because of that ability to get past that. Okay. So cholesterol, though, helps reduce the permeability to most things. Okay. And we want that. Uh, when I've read studies about young children who were put on low cholesterol diets because mothers were paranoid about having overweight children. They themselves been overweight and teased as children. They would make sure their children didn't have to go through that. They kind of went overboard the wrong direction and put them on very low fat, low cholesterol diets. And the children began showing behavioral problems and learning difficulties very young. And so, at least a few cases that read about that were published, uh, doctors caught it. Okay, and uh, they thought they caught it by thinking maybe there's a food allergy or something. Couldn't figure out what was going on, and so they had the mother track the diet. And when the 2 weeks later, the mother brings the diet in, they're like, "Well, where's the fat?" And the mother's like, "Oh, I'm not gonna feed him fat." And it's like, yes, you have to. At As children, they've got to have that. Children can metabolize fat in ways that we kind of lose that ability as adults, and uh, they're using fat and cholesterol in a fast way to build as they grow. And so this is when all the, the brain's forming. The brain's fat. I don't know if you knew that or not, but the brain is all fat for the most part. Uh, and then this cholesterol around it in the protective barrier. They got to have that as they're growing and developing. Later, as we get older, maybe not so much, but there. Progesterone and testosterone, of course, are hormones that are made pretty much throughout our lives, different amounts depending on your gender and your stage of life. Progesterone is the precursor for estrogen. And so, actually, at one point, I learned that progesterone is the precursor for testosterone as well. But I think that I have read since that's not the case. But they're related in structure in that they are both steroids. Okay, in that steroid classification. Okay, so there's your pictures, diagrams of those. You can kind of see how they're related. They have the same base ring structure. That base ring structure is your steroid structure. Okay, cholesterol. Oh, hold on here. Cholesterol, testosterone. I don't think I've got progesterone picture of it, but it's going to have that same base steroid structure. And then what makes it different is what's attached, okay, to that last ring. Your progesterone won't have the OH here. That's what makes this a cholesterol. Okay, it's the alcohol group added there. This is going to be this is a ketone unit, isn't it? Ha, yeah. Testosterone, progesterone. So you're going to see that on the progesterone. Okay. All right. Oh, we're getting close to the end, I think. Nucleic acids. DNA is short for deoxyribonucleic acid, and RNA for ribonucleic acid. And those are our genetic molecules. Oh, I have a whole presentation on those separate from this. Ooh, I I probably, gosh, do I need to give that to you? Probably so. I hadn't counted on that. Okay, well, we'll finish this and then I'll worry about that. ATP is adenosine triphosphate. It's a nucleotide with 3 phosphate groups. Adenosine triphosphate. So adenosine is your nucleotide, and you have 3 phosphate groups attached to that. And the breakdown of ATP into ADP, which is where you take one of those phosphate groups off and you just have diphosphate, is what makes energy available for cells. This is the battery of our cell. The ATP goes from the mitochondria out into the cell, carrying this battery charge, which it can then deliver when that third phosphate is broken off of the ATP unit, and that when that bond is broken, energy is held in the bond, energy is stored in the bond, so that energy is released. And it takes a little bit of energy to break the bond, but there's so much energy held in that bond that you get a lot more energy out of it than what you took to build to get into it. So then the ADP goes back, cycles back into the mitochondria. Oh, and ATP can be used within the mitochondria too. It doesn't have to leave, but everywhere else in the cell needs energy. So the mitochondria is the powerhouse of the cell, and that's what it does. Is it charges these batteries to go and deliver energy in this form. Goes back, the ADP goes back to the mitochondria. It gets recharged by having a phosphate group added onto it with that energy stored in that bond, and then it cycles back out. You may say, well, why can't the ADP just hang out there and have that next bond broken on it? Take the second phosphate off there. It can, and it does if you're starving to death. Okay. The problem is the energy released out of that bond is not nearly as much as that outside bond and that third bond. And it requires energy to break those bonds. So it's not efficient. Okay. In worst case scenarios, dying cell, starvation, severe starvation, you're dying. Okay, ADP will stay there and try to power the cell, and then AMP will too, and adenosine monophosphate. You can get that last phosphate off. What I read is that it takes about as much energy to break the bond on that last monophosphate as you get out of it. So it's really kind of desperation, active desperation, and there's not much to be gained for the cell out of it. But in the case of there's no food coming down the line, the cell will do what it can. And then as soon as food comes back into the system, if there's any ADPs trying to do this, the AMPs will go back in and get charged up fully. ADP, ATP. It doesn't mess around with ADP. Okay, ATP is the optimum battery charge situation, and that's what the body wants. So, yeah, my understanding is it's always ATP, ADP, ATP cycling, and you don't see ADP used except starvation, basically death by starvation happening. Okay. All right. That's it. Okay. I'm going to look at the test and see if I need to do the more detailed, if I need to go into detail on the nucleic acids for you. If I do, you'll see that up here as a standalone nucleic acids. Well, it'll be called DNA something or another. So this is, of course, my lecture here is designed for spring of 2020, where we are isolated off for the coronavirus pandemic. If I'm using this in future semesters and having you watch it or listen to it as a supplement, then you need to pay attention in class or to other instructions I'm giving you as to what your semester holds. But this is information that I'm giving currently to the spring 2020 class. So just I will probably try to send out an email or something to notify you, probably send out a remind to notify you that I've put up the final nucleic acids lecture if I do it. Okay, I won't burden you with it if it doesn't look like I've got a significant amount of questions from previous exams off of that. If I do, I will put it up there. If not, I'll probably just drop it out. Okay, let me see, is there anything I do want to say about that in case I don't? Well, the DNA is the blueprint for our cells, for our bodies. Every cell has its own, has everything that it needs in order to make a new you in every single cell. RNA is the messenger that takes the portions of the DNA out into the cytoplasm of the cell. It's going to bond to ribosomes out there, and it's going to make proteins. So our protein synthesis that we talked about earlier, where that the amino acids were hooked together, that's done at RNA instruction. And if I do give you the more detailed talk, there'll be a whole table and thing where we look at that. And of course, the DNA is the code that's read, and that'll, like I said, there'll be a whole presentation I'll have to go through with that if I do that. That's the main basic stuff. If you didn't already know that from, or remember that from a high school biology class, is that your DNA codes for nothing but proteins. That's all it does, just codes for proteins. And those proteins are made, and those proteins then function to turn on and off reactions and to be structural components of the cell. And that's it. The whole blueprint of life, key of life, is proteins. The coding for proteins. That was, to me, a pretty amazing revelation when it was finally presented to me in that way. It was like, wow. So I'll leave you with that.